Good morning, everybody, and welcome to today's edition of Marco's How Tuesday webinar series. This one covering Northeast Regional Marine Fish Habitat Assessment data. I'm Carl Villacoba. I am the Communications Director for Monmouth University's Urban Coast Institute, which is located about a mile from the beach in West Long Branch, New Jersey. And I work as the lead for the Marco Portal technical team. For those of you in the Mid-Atlantic and Northeast, I hope you did okay with this week's snowstorm. It was a tough bit of shoveling for us here, a good 3.5 hours for me yesterday, and feeling that this morning. Anyway, the Mid-Atlantic and Northeast Ocean Data Portals, in coordination with the Northeast Regional Habitat Assessment, or NRHA NERA, and the Marine Life Data and Analysis Team, which we call MDAT, recently published a series of maps that illustrate the relative abundance of several coastal fish species in the northeastern U.S., as reflected in repeated coastal trawl surveys. Today's webinar will highlight these data and the NERA effort it was derived from. So we'll have two presenters today in this session. First, Duke University Marine Geospatial Ecology Lab Geospatial Analyst Sarah DeLand will give us an overview of the data on the portals, how it was compiled, and show us how to access them on the portals. And next, Mid-Atlantic Fishery Management Council Fishery Management Specialist Tori Kentner will share further background on the NERA and demonstrate how to access many additional marine life map products on the NRHA Data Explorer. It's a tool that I'm sure portal users will be really interested to learn more about. Following their presentations, we will open it up to a Q&A. You're welcome to type in your questions in the chat anytime, and I can read them aloud during the Q&A. Or you can just raise your hand during that Q&A session, and I'll unmute you, and you can ask them yourselves. So just an advisory that this webinar is being recorded. You will be able to find it on the Marco Portal's webinars page and blog by tomorrow, maybe even today if I can squeeze it in, hopefully. And with that, I will turn it over to my fellow Portal Tech team member, Sarah, to get us started.
Thanks, Carl. And welcome everyone to this How Tuesday webinar. We're glad that you're able to join us, and we're excited that we've got these layers publicly available on the portal, and we're excited to show you how to use them, and hopefully they'll be useful in your work. So I'll just walk you through what we'll be covering today. I'm just going to give a bit of background on why these layers came about and the motivation behind that, and then Tori will take over and cover, more info on NERA, how the data was prepped, and the methods to get these relative abundances, and give you some info on the NERA Data Explorer insight that they have. And then I'll come back and show you how to access this data on both of the portals, show a little bit about the Marine Life Data Analysis Team site and how to access the documentation, and then get into a live demo on the Mid-Atlantic Data Portal. So there are a couple of different goals behind getting these updated data onto the portals. The first was really just to get something updated, more current. And so we had coastal trawl data previously on both of the regional data portals that covered 2005 to 2015. And now we have more current layers that cover 2010 to 2019. And part of the rationale behind that time period is that this will now match what the federal trawl data is on both of the portals, as well as several of the other marine life data sets that we've been adding to the portals. So the recently added zooplankton data covers 2010 to 2019. Some of the North Atlantic right whale data also covers this time period. So we're trying to get a lot of the marine life data more aligned into the exact same time period so that they're a little bit more comparable to one another. Another motivator for this was instead of the previous display of coastal trawl data as the individual trawls, we have now combined that data into one singular layer, and makes it easier to look at these fish species as a whole rather than as individual trawls. And Tori will get into how that was done. And then we did want to, help highlight some of the work that the Fishery Council is doing, both with NERA and with their own projects. And so helping to link both of their products to the portal and back to them. And so we've linked to NERA with these data sets. We also have shown some of the fishing effects data that they've compiled and some of the essential fish habitat work that they've been doing. And so just having better linkages between the Fishery Council and the NERA work that's being done so that we can highlight the great work that they're doing. And with that, I will turn it over to Tori to share her screen and cover the NERA project.
Great, thanks for having me. Okay, so I'm going to go through NERA and kind of how it came about to begin with. So NERA was basically came about in 2017. So our goal was how to describe and characterize estuarine, coastal, and offshore fish habitat distribution in the Northeast. So we had kind of 4 actions. So where are fish now? Where were they historically? Where do we think they're going to be in the future? Also, from a habitat perspective, what habitat vulnerabilities are out there in the area? And what is their response to climate change? Then we also wanted to look at like the spatial distribution of species habitat. So how are they using the areas, especially the offshore area? And then a habitat data visualization. So how can we make this so that we can all look at it and create tools that people can use? So we started off by going through the focus species. So Mid-Atlantic Council, New England Council all have species that were designated to manage. And then we also added species from the MAFMC, and then also 2 species, sandbar shark and dusky shark, which are highly migratory species with habitats of particular concern. So that was kind of the species we were focused on. And so what we did is basically set out to create this data inventory. So it was basically just a spreadsheet to start with, which you can see a screenshot of it there. And so we looked at all kinds of different variables in the Northeast. So we did sediment and habitat, bathymetry, water chemistry, catch data, so fisheries data, oyster reefs, SAV. We put all that into this table and then started creating these metadata pages, which you see on the right. So each data set has its own metadata page that we created. And that's just a 1-page overview. So it tells you everything you might want to know about that data set. So who collects the data, what are the time periods, what's the methodology used, any caveats, how you can access that data. All of that was put into a 1-page. So those are all housed on our NRHA Data Explorer, which I'll get into later. But basically what happened is we had this spreadsheet. We saw what data was out there, but we wanted a way to really visualize it, to really bring it all together into one place. So that's how the Data Explorer itself was developed. So that we decided to use R-Shiny because it's a really easy tool to use to create websites. And so we brought in a lot of these data sets into the R-Shiny app and then did different data visualizations with those. And so really we wanted this to be kind of like a first spot that people can go to find out what data is in the area and how they can access it. So I'll just go through, there's a number of modules in the Data Explorer. I'll just go through those briefly, and then at the very end, I'll give you a tour of the Data Explorer. So it starts off on kind of surveys and species level data. So you can look at individual surveys or all surveys combined. You can also look at individual species or a number of species combined, and you can get a ton of data visualizations. It's hard to show it all here. So again, I'll show it all in the Data Explorer later. But here's just 2 snapshots. So on the left, you see summer flounder 2000 to 2019. So this is basically just like a distribution of where this species is caught in surveys, so state and federal surveys. And that's the product that we're sharing with the data portals, which Sarah will show more later. And then on the left is just another visualization we have. So we have stage-based analyses. So this is a map of juvenile and adult distributions on one map. Based off of their maturity length. And then we also created species profiles, so basically a report for each species outlining things like their ranges, habitat, use for eggs and larvae or juvenile adults. So all of that is all on our Data Explorer. And so just to give a little more background on this particular layer, because that's what's going into the Marco data portal. So species distribution maps, we took a hexagonal grid and we put that across the entire Northeast. And then we calculated a mean abundance for each grid cell, and that's based off of the survey data itself. Because the data comes from so many different surveys and they're listed on the side there under data sources, we wanted to normalize those so that kind of the catch effort was the same across different surveys. And so we created basically a table of all of the surveys, kind of what seasons that they are surveying, gear, all of those different factors, and basically normalized all of the surveys to each other to one factor, and then used that when we were creating our mean abundance. And so on the app, you can actually use temporal filters, so things like year or season. So you can look at specific years, year ranges, specific seasons, multiple seasons. So it gives you a really great opportunity to kind of dig into this. Where's this species at different times of the year or different time periods? And in the app, the data covers 1963 to 2019. And one of our goals for this year is hopefully to update that to 2025. So sharing the data with Marco, why Marco? So Marco is a great data portal, and it really gives us a chance to really get our information out there. So it's a broader audience. And then there's a lot of context integration there. So they have a lot of great data layers on wind and shipping, conservation. So all of those can kind of be combined. And it's a great opportunity for us to kind of mesh our two products together. Another thing that you'll find in the app is these community level basis function models. And so my colleague Chris Hake put together these models. And so it's a joint species distribution model. So you see not only how species are interacting with their environment, but also how they're interacting with each other. So you kind of get a deeper layer there of those interactions. And then those models cover 20 years, so 2000 to 2019. And there's a lot of variables that go into that, but just a few are like depth, temperature, oceanographic variables. And so I won't go into a lot of that, but if you're curious, let me know, because Chris has actually done a YouTube video on this, so you can get a lot of background on his models. Another part of the Data Explorer is the climate vulnerability. So these are species habitat crosswalk. So we created vulnerability matrix for 66 managed species. And so you can see there on the left is what it looks like. So it gives you a chance to see where habitat climate exposure is high versus their sensitivity of the individual species is high or low. And so you get this nice matrix to see kind of like what habitat is most important to each species. So that's available as a visualization and also as a report. So visualization on the left and report is seen on the right. And then the app also contains a ton of publications and reports. So the modeling paper should be coming out soon. Chris is also working on an R package, as well as another optical paper, which is in development. All those metadata pages that I mentioned are all in there. We have a number of reports that we've done that are all available in there. And then also you can see just a snapshot of it on the left is our SANE trawl, or our SANE comparison. And so that gives you a chance to kind of go through each survey and see kind of what were the different methods used and how do they compare side by side. So if you're looking at any of these surveys and you want to do something similar to what we've done, you can have that background information on how these surveys could be compared. So how are we using NERA? So one way that we've used it a lot is in the essential fish habitat. So that work is just wrapping up. We're finalizing our updates to our essential fish habitat maps. And so that I think goes to the council in April for final vote on whether they want to adopt those new maps or not. But we were able to bring in all of these different layers. So not just the NIMS survey, but also a lot of these inshore surveys. So it kind of gave us a really new depth to the amount of information we were able to bring into EFH. We also use this information for State of the Ecosystem report. And then also for NOAA, there have been a few ecosystem tours that have come up that we've provided data for. A number of different projects, people have come to us and asked to use these data sets. And so when possible, we share them. Some providers don't want to share it. It's better always to go directly to the user or to the data provider. But when we can provide some information, we try to do that whenever possible. So to access NERA products, so as I said, we've got the NERA Data Explorer, which I'll share in a minute. And then now, for the first time ever, we've got products available on the Marco data portal. And so let me run through really quick a demo of the Data Explorer, and then we can pass it back to Sarah, who can go over the Marco side of things. Okay, so here is the NERA Data Explorer. The URL is up at the top there. and it's live. There's also a link to it in the PowerPoint. So I'll just run through this. So on the homepage, like I said, there's a number of different modules. There's the survey view, species view, the models, and the habitat crosswalk. So I'll start with survey view. So on the sidebar here, you can select if you want to see the entire region or just inshore data sets. I'll keep it for the entire region. You can select any of these surveys. You can see there's, I think, over 20 surveys here, both trawl and seine surveys. You can select by year, and then you can click Run Analysis. And what you'll get here, it might take a minute to load, but you'll see a map at the top of where the surveys that you selected are. So you can see the NIMS is like this dark green, and then all these inshore surveys here run all the way down to Florida. And then what the app does is it gives you an analysis. So it shows you top 20 species by abundance. And again, this is combined for all surveys. If you just wanted to see one survey, you could just select that single survey. Top 20 species by biomass. There's a list of all the species and which surveys they appear in. A cluster analysis. So this is just a little fun. It makes a little more sense if you do it for an individual survey, but basically it shows you which species are caught together more commonly. Also, if the survey has any seasonal temperature or salinity, those trends are also put into the app for visualization purposes. So if you're interested in a survey in the whole, then this is a great place to start. You can also click here on these info buttons, and that takes you to the metadata page that I described earlier to show you the metadata for each of these. And then if you're interested more in an individual species, so we'll just do summer flounder, for example, you can select that. And like I said earlier, our main focus was the 65 managed species, but these surveys capture a ton of different species. So there, I think, are over like 200 different species in here that you can look at. Any data that's available in any survey, it will show up here, even clamworm, for example. But we'll start with something more common, the summer flounder. For select a survey, so we have the NIMS bottom trawl survey. So I'm going to go ahead and just select all. So I'll turn on all surveys again, and then I'll hit Run Analysis. And if we go to our distribution map, so this is what I was describing. So this is the product that's going to go into the Marco data portal. And so what it is hex bins that show you basically like a catch per unit effort. So it's been adjusted for some of the methods that are used by each survey. And if you want to go over to season here, you can look at winter, spring, summer, and fall to see when the species is. And again, this is when the species is caught. This is only catch data. There isn't any modeling involved in this. So if no one's surveying in the summer, you're not going to see a lot of data. You can select by year, so you can do single year, or you can do year ranges. So maybe we want to turn off season, and we'll just look for the last, 10 years, 2009 to 2019. You can do that. You can grab this, drag it, see another 10-year period. If you're on single year, there's a little play button here, and it will just run through the data for each year. So there's a lot that you can kind of dig into here. And so, like I said, this is the product that we shared with the portals. So they have a subset of our species, and you can check that out. I think Sarah will share that in a minute. Something else you can look at is abundance and biomass across time for each species. So that's for each individual trawl. We have a relative abundance plot. And so this, again, is broken up into different year ranges. And it kind of shows you for each species in different salinity zones how common that species is. So summer flounder, for example, in the coastal Rhode Island, Narragansett, it's either seawater or unclassified, and you can see it's abundant to common and occasionally rare. And so that's broken down by month. So it gives you a really great way to kind of dive deep into this data and really understand what you're seeing and when that species is seen by each survey. And again, this is dependent on when the surveys are doing their surveys. The length data, I think this is particularly interesting. So on the side is the distribution of lengths for each survey. And then at the bottom is the total number of catch for each, either adult or juvenile. So adult in blue, juvenile in green. And then the map here kind of shows the trends. And so you see actually along the coast here for summer flounder, more juveniles, and then further offshore, more adults. So it just gives you a good way to kind of get a snapshot of like where juveniles and adults are distributed. So like I said, this app is packed full of reports. So here we have a species report tab in that you can look at the narrative. So narrative is like the climate vulnerability that we were talking about earlier. There's a species profile, so there's a background and life history, and then essential fish habitat designations. So we actually are working on these new source documents, so they're not in there yet, but they should be in the next year or two. So models, so this page needs a little bit of an update. Chris has done a lot of work since this was last updated, but it gives you a chance to look at kind of roughly what the models are showing. So this is, I'll just leave it on Atlantic called adult. So Chris is running both presence, absence, and count models. So you can look at a species distribution map for different seasons. There's a number of things in here. So smooth terms, so how the species is interacting with each of the covariates. Year effect, so how year's affecting it, a vessel effect, and then an optics effect. Because he's doing these joint species distribution models, you can actually see interactions between species. So here's an example. So this is Atlantic cod against all these other species. And so you can see kind of the trends, positive or negative correlations that the model is picking up. You can also see the percent of variance. So for each of these different species, this figure is showing you how important each variable is to that species. So bluefish, it looks like bottom temperature is more important than it is for summer flounder, for example. And then I think this is just variable significance. So kind of similarly, how significant is each variable to that species. And again, this should be updated in the next year or so. The habitat crosswalk. So I went over this briefly, but essentially you can choose your location, so New England or Mid-Atlantic, choose your managed species, and then you can select your age class, and then this will show you which habitat is kind of most critical to that species. So what are they most sensitive to, and what has the most like climate exposure. Species narrative. So again, this is the same report that's on the other page, but it just runs through that instead of in a matrix, just as a report. And then, like I said, lots of reports. So all the metadata pages live here. So you can see all those environmental variables. They each have their own report. So here's one on the glories, the model. So all of those are in here for all the SAV layers. That's all available in here as a one-page overview. So again, when is this data collected? What is the methods used? How do you access the data? All of that's in there. Reports that we've done are also in here. So those are there. I think the bottom trawl survey comparison, I need to check that out. Doesn't seem to be working, but the same survey comparison is working. So you can see what kind of gear is being used, what's the mesh size, what's the survey design, head rope, time of year, all of that is in here. So I think that's maybe a caveat, I should say, for the NRHA Data Explorer. We're putting all this data together into one place, but it also, I think, is important to think about, like, is this data directly comparable? Is data that's collected, bi-weekly, July through September, comparable with something that is collected, maybe only in May through November or something like that? So this table gives you an opportunity to kind of ask those questions for yourself. And then also, again, because these surveys are so variable, we have a survey data collection timeline. So what years does the survey cover? And also what months does each survey cover? So it gives you a chance to say, okay, I see we don't have a lot of data in March for most of these surveys, January, February, March, but we do have information for almost everything for September, October. And so maybe that's a good time to compare these surveys. About us, you can just read a little bit more about how this project came about. And I think that's about it. So I'll pass it back over to Sarah, and then if people have questions, I'm happy to take questions.
Great. Thank you, Tori. Let me reshare my screen. Let's see, there we go. Okay, so I will talk a little bit about how to access the data we're hosting on the portals, and I'll just use a couple quick just screen grabs before I do the live demo, and then talk about the MDAT site. I'm not going to do a demo on the Northeast data portal, but just to show you here that if you click in the marine life and habitat fish widget, it will take you to the fish page where you can select the NERA coastal trawls. They are associated with the NEFSC federal trawls, so those two data sets are associated with one another, where you can then click the drop down, pick your species, pick your season, and view the NERA layers there. This data is also on the Marco portal, and this is where I'll do the demo, so I won't spend a lot of time here, but just to highlight that, once you pull this data up, you can look at the trawl extents, and then there is associated information, and so you can click the layer and interact with it on the portal if you want more information. And then finally, on the MDAT website, we have the associated documentation for all of the different data that we're hosting. And so for the NERA data, that's under the fish relative abundance. So you can either click that to get to the web services. We have the metadata PDF available, and then we do not have a download available for the NERA data just because the survey data is a bit sensitive and we didn't want to be distributing it, but we do have a methods document that Tori and her team have developed. So I will switch into live demo mode here and start just with the Marco data portal site. And so if you scroll down here to browse the data portal, it will bring you to this tab where you can say view maps. We'll let that load for a second. So under this data tab, we have different categories that you can choose from in the table of contents. And the NERA data, as well as a lot of the MDAT data, is under the Marine Life Library species specific category. So if you click this header, it gives you this drop-down menu of all of the marine life data that we have at a species level. And so similar to the Northeast portal, we have both the NEFSC, the federal trawl information, as well as here we have the NERA and the coastal trawl information. So those data sets are being kept separate. I will point out, though, that the NEFSC species, while there are more species under this tab, all of the NERA species are included in the NEFSC species, and they both cover 2010 to 2019 and fall and spring. So they are very similar to one another, and we've tried to structure that deliberately. So if you go under the NERA abundance, you can click to add the coastal trawl extents to your map. And if you click this little info button, you'll get information on each one of the different coastal trawls. I won't scroll through because there's a lot of trawls and it's a lot of information, but you can scroll through and see all of the different trawl descriptions. And then if you go here to species abundance, again, if you click information, you can see a general layer description on what this data set is showing, the units, etc. And we do have a link to the NRHA Data Explorer to help with that connectivity between the two sites. And then you go down here and you can search for a species if you have one in particular. There's also a search function up at the top if you don't even want to go through all the different tiers in the table of contents, but you can scroll through the 14 species that we have NERA data for. So like Tori said, this is a subset of what they have on their site, but we wanted to pull out some coastal species for this time period specifically so that they are available to portal users. So for this example, I will do bluefish relative abundance for the fall. And once I've added this to my map, I can go to this active tab where I have both the coastal trawl extents and then the bluefish relative abundance. I can go here to adjust the opacity. I'm going to make this data just a little bit darker while we're looking at it here. And since that kind of obscures the coastal trawl extents, I'll pull those on top. Once I have this data kind of set up the way I like it, you can see if you click in the legend tab that we've got, these are relative abundance, so they go from low to high, and we've got the different trawl extents. I know that these are hard to distinguish. It's a lot of trawl data in a small area, but if you click on the map, you are able to get information on what trawl is in that location. For the NERA symbology, we also did want to make sure we distinguished between what is of 0 value versus a no survey value. So if I scroll south a little bit, you can see here's some examples of if I click on this hex bin, the mean catch per swept area is -999. So that's the no survey value. And so we wanted to make sure it was clear where no actual surveys were conducted versus a hex bin like this one where there were 2 surveys, but there were no bluefish caught during this season and this time period. So that is something that we wanted to make sure was distinct for portal users. Okay, so I will also go through and show, pan a little bit north again, but if you click on any of these hex bins, you do get the info, like I said, of what trawl extent overlaps with that. So here we've got the NEA bottom trawl. And then if you scroll down, this is the information associated with the NERA data. So some just kind of generic information on the hex bin ID, what that common species name is, and the season. And then as you scroll, we did want to incorporate, because we're for the first time incorporating multiple coastal trawls into this data set, showing what trawls are in each hex bin. And so for this hex bin in particular, the Maryland bottom trawl and the Neomat bottom trawl are the 2 surveys that are providing information to this data point. And for those 2 trawl sources, there were a total of 70 surveys that contributed to this point. And then if you scroll down even further, we have an attribute for each of the coastal trawl surveys that are included in the NERA data set, where you can find exactly how many surveys were included. So here, NEAMAP contributed 10 of the surveys to this data or to this hex bin, and then the Maryland bottom trawl contributed 60 surveys. We scroll back up. You can see here both the actual value, so the mean catch per swept area that was calculated by NERA, and then the relative abundance, which is what is being symbolized on the map here. So that's all the information that we have included in each of our attributes. One of the other reasons we did want to make sure those trawl sources were included is because NERA has on their site so much information about these different trawls. So then knowing that, it's the Maryland bottom trawl, like Tori pointed out, you can go back and inquire on their website about this trawl's methods and like their survey methods so that you can get more information about how this data was collected. And so it gives you the tools you need to get more information if that's something you want. Okay, and now I will go back to this table of contents here, and I will just show you how to look at this data in the data catalog. And so if you're looking at this layer information again, there's this button here that says Read More. So once you click that, this takes you to the Marco data catalog. And so under this NERA entry in the catalog, again, we've got this layer information on just a general description on this data and how to access the NERA app, what the data source is, a little bit more information on the units, and then the species that are included. From here, you can also click to get to the metadata as well as the methods document that the NERA team developed. So I will click both of those and just do a quick scroll. So here, let this load for a second. Oh, there we go. So here is the metadata for these documents, and it allows you to see, the data contributors, a little more description of the background of both of the data providers, MDAT and the NERA teams, how to cite this data if you use it, and some of the geographic information. With the NERA methods document, we'll scroll us to the top. You get a description on both the NERA project and their background, the methods used for the bottom trawl surveys and how they created the swept area calculation and ultimately got these species distribution maps. And we wanted to make sure we had this just because what is shown, as you saw from Tori, what is shown on the Marco portal and the Northeast portal is slightly different than what's on their website. Again, we did that relativized abundance. We have a subset of the species, and we have a particular time period, 2010 to 2019, whereas on their site, you can really customize that. And so we wanted to make sure it was clear that, these were a little bit different, and so they have specific methods associated with them. Finally, we have field names and descriptions. And so for any of the trawls, if seeing these kind of abridged names is confusing, we have descriptions in this methods document and just definitions for what each of these attributes are. And then finally, If you need another way to access this data, that is all on the Marine Life Data and Analysis team or the MDAT website. And so if you scroll down, like I showed in my screenshot, we do have this table of different products that we provide. And so here we do have the web services to the relative abundance data. Like I said, there's no download available, but we do have these web services, so you are able to, work with that data in ARC Online or pull the web services into your own projects and still work with them despite them not being available for download. So I think I will stop there. That was a lot of information, and we can take any questions.
Thank you so much. That was really informative, and you showed me some things I'd never considered and didn't know there. And so, yeah, I would welcome people to um, type their questions into the chat or raise their hand. We do have one question that came in while Tori was presenting, which seems to be a bit of a technical one. This is exciting to see. This is from Catherine Ford, NEFSC, who writes, "This is exciting to see. Thank you. NIMS runs many trawl surveys. Can the name be changed to the proper name NIMS NEFSC Multi-Species Bottom Trawl Survey? Even NEFSC Bottom Trawl would be more indicative." And then she wrote shortly after that that she did have to jump to another meeting. So if that's something you want to touch base with her offline, I can, uh, if you don't have her email, I can get that for you.
Cool. Thank you. I can do that.
Um, One question I had, Sarah, well, probably for you, and then Tori might have some thoughts on it as well. Um, so for people who don't know, Sarah and her team at Duke are responsible for the lion's share of the marine life data that we have on the portal, in particular, marine mammals, birds, the fish data, and all, most of that MDAT data is federal waters, so it starts at that 3 mile mark, moves out. Where this really was the first time for a lot of these species that we had anything that went into Chesapeake Bay, Long Island Sound, Delaware Bay. And so my first curiosity when we got this was to turn on some examples for example, black sea bass of, NERA 2010 through 19 with the MDAT version, which was NEFSC. And I'm just curious if you have any thoughts on the wisdom of that. Is that something that is a good or a bad idea to do, or caveats that you think users should consider if they do that?
Yeah, I think part of the motivation was that these can be more comparable. They are still different. The NEFSC data is biomass, whereas here we're doing relative abundance. But ultimately, the idea is that this coastal trawl data does kind of fill that gap, as you mentioned, Carl, between those the federal trawls, which go 3 miles offshore or beyond, whereas these coastal trawls cover, as it sounds, the more coastal areas and the inland waters. And so it's definitely made to fill a gap that we had. And we did have coastal trawl data, but like I said, it was all very piecemeal and it was just each individual survey. And so on its own, it's a great tool for the more inland fish data and can cover some of those gaps that the NEFSC data has. But they are complementary. They are, like I said, not directly comparable, but complementary. So, they are meant to be used with each other, but with a bit of caution and awareness that what you're seeing is slightly different units. And so they're not going to be one-to-one comparisons. And Tori, chime in if you have other thoughts as well, because the NERA report, or yeah, the NERA Data Explorer does include the NEFSC trawl data, if I'm remembering correctly.
Yeah, that's right. Yeah, we have inshore and offshore on there. And then also, you mentioned the relative abundance that we did for the portals, and that actually should roll out with the new data sets through 2025 that we're hoping to finish up this year. So even the portal should be more comparable to what we have on the Data Explorer in this next year as well.
That's great. Thank you.
Jay, you look like you're about to ask something.
Yeah,
You
well,
physically raised your hand rather than that's good.
it's easier for me to find this hand than the digital one. It seems to keep moving. Just a quick observation. Well, we just historical perspective, we thought this would be a clever idea to do about 15 or 20 years ago. And when the portal was first being launched, what if we could combine all the different trawl surveys and make some sense out of it? We spent a bit of money having people look into that and decided it was completely impossible and we shouldn't waste any money trying to do it anymore. Uh, so it's really exciting to see what you guys have pulled together with this. I mean, the site, it's the nearest site itself is such a rich treasure trove that I am just still trying to understand and scratching the surface of in terms of like I don't know all the things that can be it's sort of a it's a rich thing that can be mined for a lot of different types of derived products, I think, and papers. The I'm still struggling a little, just trying to figure out how to talk correctly about all of the data products. And I when we were struggling with like trying to combine, apples and oranges early on, and you guys figured out some clever ways to kind of normalize abundance, normalize catch, and normalize might not even be the right word, between the different surveys. And then and we have those hex bins. And then there's all the work related that went into the EFH, the EFH that's going into the each omnibus amendment. That's more of a model derived, continuous data, predicted densities based on the environmental covariate fits and all that. Can you just riff a little bit without getting, for to talk about sort of the two different, I don't even know if it's right to say two different approaches, but just those different types of data products and some of the challenges and opportunities around them. That if that gives you enough to go
Yeah,
on.
for sure. Thanks, Jay. Yeah, I think you make a really good point about the challenges of combining these data sets together. It was something that we thought a lot about as our team, and I still would add caveats to it. That's like you can decide for yourself if you should do this or not. I think, we did our best, as you said, to normalize these data sets. So we looked at as many different factors of the trawl as we could, like down to like the mesh size, head rope, foot rope, all those things. Took all that into account to try to create this like um, unified unit that we could use to kind of compare these. But still, there's so many things, seasons, the years, all of that, that you have to take into account. So we did our best with it, and I like what we came up with, but I think there is always room for improvement on it. So those hex bin maps are kind of just survey data. And then what Chris Hake did is he then took that survey data, but not all of them, only NIMS, Massachusetts, Maine, New Hampshire, and I think NEMAPS, and he put those into a model with a bunch of different covariates, and then that was his joint species distribution model. So we kind of used that to do our EFH designations, and then we also, because that's just mostly offshore, we created another product which you guys haven't seen in this presentation, which is like an inshore species distribution that kind of is estuary based. So subsets the estuaries into smaller pieces, and then if the species is seen in those estuaries, kind of combining that into like an estuary species zone, if that makes sense. So I don't want to go too far into it because I didn't share it here, but so both of those were combined, and so all this is kind of coming out of NERA and then being fed into other products like EFH or any I've had a number of different organizations say, oh, can we have like a subset of this data for this project, or can we visualize it in this way for this other project? So the NERA data is really just foundational trawl survey data from all these different inshore surveys, offshore surveys, and then it's kind of being fed and distributed into other projects. Or our advice is always go back to the original data source and get the raw data, just that what you're getting and not like what we processed. So I don't know if that answers your question, Jay, but hopefully.
No, it does very well. Thank you. And if Sarah has anything to add, I'd be curious in that too. Like for me, it feels to me like in a perfect world we would have like such high sampling density we could just interpolate grids of the density of critters and we wouldn't even need to worry about covariates and models because we would just have such great sampling. Of course, that's a total fantasy. It'll never happen. And my default, like I tend to trust, just for what it's worth, I tend to trust the sort of the covariate based modeling more than the map the density of everything method of a snapshot picture. And I feel like that. I'm just curious if you guys feel like that's the way the world's moving, and too in terms of because I feel like on the portal right now we have both types of products and we're kind of and I don't know that we or our users fully understand as well as they could sort of the pros and cons and caveats with those two basic types of products.
Yeah, I think that's a really tricky thing to answer, and I think what you said is right, that really in an ideal world it would all be just sample based points or combined points and interpolations. But the sampling's so dense and the data's so robust that you don't really need to do a lot of that interpolation and then those models are no longer really required. But unfortunately, we don't have that level of sampling, and so those models tend to cover a lot more of the area that people are interested in, and that's where they kind of fill that niche. But there you still have people who are very skeptical of a lot of modeling efforts, and it's kind of a black box for a lot of people in that, yeah, sure, you know what covariates are going into it, but like how are they weighted? How are they being used? And all of that information is, I mean, it's out there and you can kind of deconstruct it, but it's a lot to take in and to understand. And so I think the reason we try to have multiple types of data on the portals is to fill both those gaps of, show what that sampling effort looks like, show what that data is, and then let users identify where those gaps are, or if there's gaps in the areas that are of interest to them, and then hopefully also have model data that can help fill some of those gaps. But, you know, we do what we can with the data that we have, and we try to put the best, available models out there when we do model data. But people have critiques no matter what, and so you've got to make the best of what you have.
Thanks, that's very helpful. The other advantage to the modeling approach, I mean, I guess it's kind of a duh, but in a very dynamic climate shifting ocean, um, the estimated distribution updates to the quality of the latest ocean model as opposed to the quality of the survey that you did in 2017 or whatever.
Who else has a question or thoughts or comments? And that could also be if anyone has a question about the portal in general that hasn't been covered, I can hopefully take that.
Well, if I may, since there's a little lull,
Go for it.
one thing that I think is really interesting and cool is that the data was worked up for so many species that people have never heard of that are considered bycatch in the or it used to be coded as trash in some of the early trawl surveys, the stuff that you're not trying to catch. But and as I know, Sarah and MGEL shop is going more and more into joint modeling, which I'm just still understanding what that means, but it's the model is not just responsive to the environmental conditions, but also responsive to the presence or absence of other critters or other specific taxonomic communities. And that seems like that's really the frontier, a really important frontier as well that this project is pushing into.
Yeah, I think a lot of the work that Tori and Chris Hake that she mentioned are doing is really great work and really useful products. And that's why we wanted to have this webinar to give Tori the opportunity to show that NERA Data Explorer because there's so many resources on there of more information than just the static layers that we're showing on the portal. And so want to make sure that, people are aware that yes, this is a subset of what NERA has done, but it's not all of what NERA has done. And that they have a lot more resources that people can access and make use of.
Right, right. Just having all that survey information in a releasable way in one place is so huge.
Hmm.
Do you I'm struggling with trying not to hijack this call to talk about something I care about and I'm interested in, which is trying to develop spatial data to represent the fish habitats of concern identified by the Atlantic States Commission. And so I'm um, I'm hoping that we can make some progress on that front in 26. And that would include using, to some degree, Tori's grid, the 1 kilometer inshore grid that has a lot of attributes for basic oceanography and topography. So anyway, I have a concept note I wrote about that with a lot of help from others, and we're going to be trying to move that through the uh, Commission's Habitat Committee process for potentially become a new project that I could spend a fair bit of time on this year doing like project management type of function on. Um,
Well, yeah, Jay, I'm curious to hear what kind of feedback you get on that. Sounds like a really interesting project.
yeah, yeah. I mean, with the final vision being, you know, it won't be like this, but like if imagine, the habitat map of our dreams, which would include the finally vetted and approved EFH omnibus information and all the other official habitat designations that haven't been changed from HMS and critical habitat from ESA, um, areas identified under MMPA and so on. Then, but if it was extended to include the inshore all the way up to as high as a yellow eel will go in the little step across streams up in the hinterlands, all the way down through all the diadromous reaches for the different life stages of diadromous fishes, all the way from Maine to Florida, and you could drop a polygon or a pin anywhere on the map and immediately get a list of its critical habitat, whatever it is. It's larval habitat. This data center intake line is right next to fish habitat of concern. That's a larval habitat for sturgeon or whatever. Um, so I feel like there'd be a use for it.
Yeah, I mean that does sound like the next evolution in, NERA to EFH to like a all-in-one comprehensive product.
Hmm. Thanks for letting me hijack to give my soapbox on this. I'll shut up.
Thanks, Jay. We are about 5 minutes to noon. So any final questions from anyone on the call? Seeing none, I will thank everyone who joined us today. And thanks again, Sarah and Tori, for these great presentations. And just one more reminder that I will have a recording on this or of this posted on the portal within the next 24 hours. I realize probably a lot of people had school closings and work closings and stuff today and couldn't make it, but that's what recordings are for. And so look out for that. And thanks everybody again, and look forward to seeing you soon.
Thanks,
Yeah, thanks for
guys.
having us. Thanks for tuning in, everyone.
Thanks.